Hello, everyone, and welcome to today's webinar titled Advanced Solutions for the Development of Gene Therapies. This webinar is part of the Cell and Gene Therapy Virtual Event Series 2024, brought to you by LabRoots and is sponsored by GenScript. For more information on our sponsor, visit genscript.com. I am Israel Gonzalez of LabRoots, and I'll be your moderator for today's webinar. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by both communicating with each other using the group chat and by submitting questions during the presentation. Now let's get started. I'd like to introduce our speaker, Rumen Bogov, Head of Segment Marketing at GenScript USA. You can view Rumen's complete biography below. Rumen, you may now begin your presentation.
Hi, my name is Rumen Bogov, and I'm Head of Segment Management in GenScript. It is my pleasure today to introduce you to the advanced solutions for development of gene therapies by GenScript. So this is the agenda for today. And here a little bit about GenScript first. So GenScript is a global company. We operate in over 100 countries. We have over 200,000 customers worldwide. We have over 6,000 employees, more than 800 R&D staff. We have many patents and patent applications, and our customers have published using our services and product over 80,000. We have over 80,000 references. We operate globally, as I mentioned. We have multiple manufacturing sites. We have 2 manufacturing sites in US, one in the East Coast and one in the West Coast. We have multiple manufacturing sites in China. We have manufacturing site in Singapore, and we have commercial offices around the globe. My presentation today will be focused on our capability as CRO, CDMO, and products company. So basically, GenScript Group consists of several entities, and my focus will be on the entities on the left side. But I'll very briefly introduce the entities on the right side because they're also part of the GenScript Group. However, they work in different areas than what I'm focusing on today. So Bestzyme is our synthetic biology company. They have very strong capabilities in synthetic biology, and they focus on human and animal food supply. And they have very large-scale capacity to produce enzymes, proteins, and stuff related to these industries. Legend Biotech is an innovative cell therapy company. It works on delivering cell therapy companies for a range of disease. About a year ago, they launched Carvykti for multiple myeloma, working with Janssen. And they have advanced pipeline of developments in other disease areas like leukemia, solid tumors, infectious disease, and so on. So the entities on the left, the Life Science Group, is our division that contains the CRO division and also the product division. And they deliver a lot of innovative and reliable services and products for therapeutic developments. We use a lot of automation in our CRO division to increase our efficiency, and we provide cutting-edge services and products. And we have also a lot of products for automation, automating plasmid purification, automating protein purification, antibody purification, and so on. Our biologic CDMO division, ProBio, focuses on GMP production, and they have very large GMP capacity. They have very strong regulatory experience, and they're one of the leading biologic CDMOs service providers in the world. So a little bit about the gene therapy pipelines around the world. But first, of course, when we refer to gene therapy, we very often talk about in vivo gene therapy and ex vivo gene therapy. In vivo gene therapy is when the editing is done inside the human body using either viral reagents or non-viral gene editing reagents. For ex vivo gene therapy, usually cells are extracted either from a patient or another donor. They're genetically modified using lentivirus or non-viral gene editing reagents, and then injected back into the patient's body. For the purpose of today's talk, my focus will be mostly on the in vivo gene editing. I'll passingly mention some of the things that we have, also some of the products and services we have for the ex vivo gene therapy. So looking at the pipelines of therapeutic developments around the world in the gene therapy area, cell therapy, and RNA therapeutics, this is data from AGCT. They're showing that there are about 4,000 therapies in development, ranging from preclinical to pre-registration stage. And about 2,000 of those therapies are about 2,000 of those therapies are either genetically modified cell therapies or gene therapies. So very strong pipeline, a lot of activities in the gene therapy area. Then if we look a little bit more in detail on the preclinical stage, there are many therapeutic developments, over 1,400 in preclinical stage, maybe about 600 or so in phase 1 to phase 3 trials, and then a few in pre-registration. So again, very strong pipeline of over 2,000 therapeutics in development for the gene-edited cell therapy and gene therapies. So looking a little bit at the process for developing gene therapy, there are a lot of different reagents that can be used, and the typical methodologies are either using viral gene editing or non-viral gene editing. They have certain advantages and disadvantages. So for viral gene editing, for example, one of the advantages of viral gene editing is that it provides very high efficiency in delivering and expressing the genes. So it's very efficient at doing that. While the non-viral gene editing is not as efficient at delivery and editing, however, is considered much safer profile. Same thing if we look at the safety. If we look at the safety profiles of these methodologies, again, the viral gene editing is considered creates more concern around immune response, more concerns about potential cancer issues, and so on in the long term. And the non-viral gene editing doesn't have a lot of these issues. However, again, efficiency is still a concern. Efficiency still needs to be improved, but it's very attractive because it's better safety profile. If we look at the packaging capacity of both methodologies, viral vectors have more limited packaging capacity because the genome size is defined for each of the viral vectors. Well, for the non-viral methodologies, packaging capacity is not an issue. You can pack a lot of genomic material and deliver it. However, the bigger the cargo, the harder it is to efficiently deliver it and achieve the editing. So if we look at specifically the non-viral gene editing technologies, through the times, different technologies have been developed, and zinc finger proteins were one of the first genome editing technologies being used. They have, basically for the purpose of the gene editing, zinc finger domains are assembled that bind to specific DNA sequence, and they're linked to FOC1 endonuclease. And then basically that way they bind to the DNA and make a cleavage. For the purpose of the editing, you have to develop this protein complex that contains these zinc finger domains and the FOC1 endonuclease, and then you have to assemble a dimer bridging between the two sections of the proteins. And that's a little bit challenging to do. Also, for every DNA sequence, you have to generate different enzyme, different protein complex to do the editing. For TALEN editing, you have similar situation where you have to create these TALEN sequences, tail sequences that bind to specific DNA sequence and link them to the FOC1 endonuclease. However, in this case, you don't have to link both sections of the complex. They can be independent. Also, tails are better researched. It's easier to find the right sequences of tail domains to link them together and bind to the specified DNA sequence. However, the need to develop new tail sequences for each DNA sequence is still a challenging task. CRISPR-Cas9 was developed later and delivered a lot of conveniences in how gene editing is done, because the affinity of binding is determined by a relatively short RNA sequence, and this RNA sequence can be used with the same enzyme over and over again for different DNA modifications. That makes it very convenient and easy to do a number of different modifications very quickly. And then if we look in a little bit more detail in the CRISPR gene editing, basically using the CRISPR-Cas system, you have sgRNA that basically determines the sequence of the DNA that is going to bind. You have the Cas enzyme that binds to this sgRNA and cleaves, and you have double cleavage here. And then during the repair process, you end up with the modified genome. This is very convenient process, is very flexible. However, it has some challenges with efficiency and some challenges with specificity. So later on, base editing was developed in order to improve the specificity and efficiency of the editing. In this case, instead of Cas nuclease, Cas nickase is linked to a deamidation enzyme, deamidase. And this deamidase can basically deamidate the A and the C, and you can have A and T converted to G and C, and A and G and C converted to A and T. For the prime editing, you have usually nickase linked to a reverse transcriptase, and then the nickase cleaves the DNA chain. The reverse transcriptase synthesizes the DNA chain according to the RNA template used for the binding and defining the reverse transcription template. And then during the repair process, you achieve the gene editing. So these methodologies have certain advantages and disadvantages. And if we look at the advantages and disadvantages for these methodologies, let's say for the Cas9 is the most flexible one. It's relatively easy and straightforward to do. It gives you ability to do deletions, insertion, modifications. It's very easy to use, but it has moderate specificity, and the efficiency of the editing is not that great. Base editing has better specificity and is very efficient at converting the single base modifications. However, the disadvantages is limited to single base editing, and only 50% of the disease of the conditions are due to single base mutations. So it cannot be used for other modifications that need to be done. And prime editing was developed to eliminate some of these shortcomings for the base editing, because the prime editing can give you more flexibility. You're able to modify multiple bases instead of single base, and you can do some insertions and deletions that are of multiple nucleotides. You can go up to 50 nucleotides in your insertions or deletions, and it's more flexible than the base editing while still being very specific. However, both of them, base editing and prime editing, have the disadvantage that you essentially assemble this protein complex that is relatively large. So the nickase with either the deamidase or the reverse transcriptase, they're relatively large complexes that are harder to transfect. If we look at the viral methodologies being used for gene editing, there are several different viruses, but these are the most popular ones. AAV has become basically the workhorse for gene editing, is because of its ability to efficiently modify the genome. It's also has very good safety profile. It's not, it doesn't integrate into the genome, and it has relatively low immunogenicity. The limitation of AAV is that it has relatively small genome, 4.8Q bases, and that limits how much cargo can be delivered with the AAV. The other viruses, retrovirus and lentivirus, they're mostly used for ex vivo gene editing because they have a little bit lower safety profile. They integrate into the genome, therefore they're considered more, there is more concern about potential cancerogenic effects later on. That's why they're normally used ex vivo instead of in vivo. AAV also has very well-established tropism selectivity. Looking at different AAV serotypes, one can be selected that can focus predominantly in certain organ or certain cell type. And because of this very well-established selectivity, is used a lot into the gene therapy area. And GenScript can offer a complete solution from beginning to end for gene therapy development. Basically, starting from target discovery, we have a number of services and products that can support target discovery, a number of services and products for target gene validation. For lead generation optimization, we can support either viral vector-based methodologies or non-viral-based methodologies, predominantly CRISPR. For preclinical work, we have the corresponding quality requirements for the different services and products that we can meet, and for R&D work. And then for clinical work, we have GMP manufacturing, cGMP manufacturing for the different components that are used in different services that we offer for the clinical work. So if we look in a little bit more detail at the target discovery and validation, what solutions we offer. Basically, target discovery very often starts on the genome side. So a lot of sequencing is done, a lot of qPCRs, looking at up regulations, down regulations. We can support the development of qPCR reagents, primers, probes, labeling the probes. Next-gen sequencing, we have enrichment libraries that can help you target your sequencing better to specific regions. CRISPR gene editing, we have full line of CRISPR gene editing services and products that can help you with knockouts and so on. For protein level gene discovery and validation, target discovery and validation, we have a lot of protein-related services, protein expression service, antibody development services for immunodetections, full line of Western blotting products, and cell-based assays for cell lines for cell-based assays for cell-based work. So we also have a special semiconductor technology, Gene Titan, that allows us to develop oligo pools very quickly and efficiently. So we can, on one single chip, we can synthesize 8 million oligos at the same time. And that makes us very efficient at developing various oligo pools and various libraries. So for example, we have offerings of developing gRNA libraries, and we develop them by synthesizing number of oligos on a semiconductor chip, then using PCR to amplify them, then cloning these oligos into the plasmid, and then we can deliver the library either as a plasmid library or we can deliver it as a lentivirus, package it in a lentivirus. For immunodetection, of course, a lot of antibodies are needed. Immunodetection techniques are very popular for like Western blotting, flow cytometry, IHC, ELISA, very popular for target discovery and validation. And we have the ability to develop polyclonal antibodies, monoclonal antibodies, bispecific, single domain antibodies, SCFVs, VHHs, and we can develop custom ELISA kits. Here I have highlighted one of our technologies, which uses basically for developing rabbit monoclonals. Rabbits are well known for giving very high affinity and avidity antibodies. So basically we can do single cell antibody discovery, immunizing the rabbit, extracting PBMCs, enriching, then running them through the Beacon single cell screening system, detecting the clones of interest, sequencing them, and then we usually generate 4 or 5 recombinant antibodies and deliver small amounts to the customer for testing. So we can very quickly go from immunization all the way to recombinant antibody using this technology. For Western blotting, we have full line of products. As I mentioned, we also have a products division. Starting from gels for electrophoresis, we have very fast running, very high resolution gels that can be used for protein separation. Then we have a number of devices related to electrophoresis and Western blotting. We have automated fast staining system that can allow you to automatically stain the gel after electrophoresis and visualize your bends. Or if you're doing Western blotting, you can use our eBlot fast transfer system to transfer the proteins efficiently to the membrane. And then we have the EasyWest light device that can automate the detection of the proteins on the membrane, basically adding the blocking reagent, the wash reagents, primary antibodies, secondary antibodies, to automate the detection of the Western blotting membrane, so increasing your efficiency. We also have very nice automated system for protein and antibody purification, AMAC-SA. It can purify 12 samples at a time, up to 50 milliliters per each sample, and it performs affinity purification using magnetic beads. So any recombinant proteins with his tag, GST tag, flag tag, and so on, and also antibodies can be purified easily on this system. We also have a lot of services for cell line development. We can do customized CRISPR knockout cell lines, whole genome CRISPR deletions, customized CRISPR knock-in cell lines. We can do screening on and off-target services. We have our cell power gene constitutive expression service or inducible expression service. So we can develop many custom cell lines for cell assays. We also have very large portfolio of off-the-shelf cell line products. These off-the-shelf cell line products are monoclonal cell lines, basically starting from a single clone, and we can deliver them as multiple species. We guarantee their performance, and we do very stringent QC of these cell lines. And their focus, our portfolio is focused on GPCR cell lines, Fc receptor cell lines, immune checkpoint cell lines, and we have some COVID-related ACE2 and spike protein cell lines. So looking at our solutions for lead generation and optimization, we are one of the biggest providers of gene synthesis services, and gene synthesis is fundamental to gene therapy developments. Pretty much every gene therapy in development uses at some point of time gene synthesis. We are the largest gene synthesis provider in the world. We started providing gene synthesis in US in 2008, and we have many, many customers. We have delivered over 3 million genes to our customers. We have 99.95% success rate. We have very large capacity to synthesize genes. We can synthesize 200 million base pairs per month, and we have global production network with multiple locations for gene synthesis. We have excellent codon optimization software that uses over 200 parameters to optimize the sequence for your purpose, and it's free to use to our customers. We can synthesize your genes as a single fragment or the gene brick or gene fragments, essentially. So very large genes up to 200 kilobases can be synthesized, but these very large genes, sometimes in the 100 to 200 kilobases, is much more efficient if they're synthesized in fragments. So we offer the gene brick service to synthesize them in fragments for you, decrease the timeline and the cost associated with it. And we have a very nice cloning kit that allows you to later on assemble those fragments back into your plasmid vector. We also have the VectorArk and CloneArk storage systems. So if you are doing using our services, we can store your clones or your vectors, and we can store them for free, and you can reuse them later on for your subsequent orders with us. For plasmid services, we have been doing plasmid development and purification for a very long time. We have the full offering to support discovery, preclinical work, clinical work, and even commercialization. We can offer basic research-grade plasmid development and purification, industrial grade with a little bit higher quality and more strict QC, preclinical grade, CMC grade plasmids, and then basically supporting with phase-appropriate GMP quality. We can support your clinical trials and then your GMP production. If you prefer to purify your plasmids yourself instead of using our services, then we have a very nice automation system for plasmid purification, AMA4. This instrument is very flexible because it uses one controller unit, and multiple purification units can be connected to this controller unit. So you can basically, if you have low throughput initially, you can buy one controller unit and one purification unit or two purification units, and then later on you can add more purification units up to 4. Each unit can purify 6 samples at a time, so you can start with 6 to all the way to 24 samples at a time. The instrument is very flexible, can work with range of culture volumes from 50 to 200 milliliters, can purify up to 1.5 milligrams of plasmid, basically maxi prep or low mega prep scale. And runtime is a little bit less than 2 hours and produces transfection grade plasmid with very low endotoxin level, high supercoat ratio, and it's very easy to use. All the consumables are packaged into our cartridge. You just insert the cartridge into the instrument. You add your cell pellet. You add some methanol, push a button, and 2 hours later, you have your plasmids purified. So very nice automation solution for somebody that prefers to purify plasmids in-house and has significant number of them. We can provide full service AAV engineering, starting from codon optimization. As I mentioned, our GeneSmart codon optimization software can use over 200 parameters for codon optimization. And if you are synthesizing, we can synthesize single fragment or a gene brick. We also have very large ORF cDNA collection that you can use if you would like to get the clone from there. Then we can do a lot of engineering on the genome side for the vector to optimize with target-specific promoters and enhancers, transgene expression regulations, and so on. We also have guarantee. We have AAV ITR guarantee service where we can guarantee your the integrity of your ITRs. And then we can also work with you on capsid engineering to make sure that your capsid is engineered in a way to target the specific cells or organs that you're trying to focus on. And we can do mutagenesis service, or we can provide mutant libraries, or we can provide oligo pools for your development of mutant libraries. And as I mentioned, our Gene Titan semiconductor technology is very efficient at developing oligo pools. And these oligo pools, we can use them for our gene fragment service when we have to synthesize very large genes in multiple fragments, or they can be used for developing precision mutant libraries for various optimizations of your plasmids or viruses. Here is just a reference paper from a customer. They used our oligo pool services to develop library to optimize their AAV5 virus to focus on the liver. And they successfully did the mutation work, mutagenesis, and achieved very high concentration of the virus in the liver. For CRISPR reagents, we can offer a lot of reagents and services from beginning to end, from discovery to preclinical development and clinical development. We have several grades of sgRNA depending on your requirements. We have multiple templates, single-stranded, double-stranded, and circular double-stranded DNA, and we can offer them in the appropriate qualities with the appropriate quality requirements for discovery work, preclinical, and GMP work. So the different templates that we offer are our gene-exact single-stranded DNA. Single-stranded DNA is very good for minimizing cytotoxicity. It gives you very precise knock-in with very little off-target effects. However, it's limited to 5 kilobases of length. So if you have a larger DNA payload, you may want to switch to some of our other templates. GeneWant double-stranded DNA is suitable for inserts up to 10 kilobases. The two is double-stranded, but the two ends are linked together, so they are closed ends to increase the stability. And this template is also very good for scale-up manufacturing for larger quantities. And then we have the gene circle double-stranded DNA. Basically, we have miniaturized vector with no antibiotic resistance genes, removing all the other genes from the fragment, decreasing the backbone to 400 and something base pairs. And this template is very good for inserts of up to 50 kilobases, and it increases the knock-in efficiency by about 30%. It's very stable and very high knock-in efficiency template. mRNA is becoming more and more popular as a methodology for delivery the gene editing reagents, and we can provide service from beginning to end, starting from codon optimization all the way to IVT mRNA reaction. Basically, as I mentioned, using our codon optimization, we can optimize using over 200 different parameters. Then we can provide the gene synthesis either as a single fragment or multiple fragments. Then we can prepare the plasmid and mass produce it. We can also linearize the plasmid for the IVT reaction. If needed, we can do enzyme production if some specialized enzymes are required. Then we can synthesize the mRNA, either linear mRNA, circular RNA, or self-amplifying RNA can be synthesized using our IVT production capabilities. And then in the end, we can package this mRNA into lipid nanoparticles, and we can optimize these nanoparticles for delivery for the different organs and different cell types. If we look at the different RNA formats, basically linear mRNA has been very well established methodology, is being used a lot because of its low immunogenicity. However, it's not as stable as the others, so it would be degraded relatively quickly. So the other options of formats, self-amplifying or circular RNA, they're much more stable, and they can produce much larger amounts of protein. They do have a little bit higher immunogenicity, especially the self-amplifying RNA. However, it allows you to minimize the payload that you're delivering, so that can compensate for the little bit higher immunogenicity. And if we look at performance in cells, we transfected here EGFP RNA into HEC293 T cells, and looking at the performance of these different template formats. mRNA, of course, you can see that at 72 hours, the intensity of the signal starts decreasing because of degradation, while the other two formats are more stable. So circular RNA produces more protein, and also you can see at 72 hours, the signal is still increasing. And same thing for the self-amplifying RNA. You can see that the signal is stronger at 72 hours, or at least similar to the previous one, and it's much, much higher than the other two formats, even at half of the amount being delivered into the cells. So we have also, if you're interested in using mRNA for gene editing, we have mRNA off-the-shelf mRNAs of ESP Cas9, circular, we have linear mRNA, we have circular mRNA, we have CPF1, Cas12 mRNA, we have the prime editors P2 and P3 mRNA. We have other mRNAs that you can use for other proteins. Feel free to look at our website and see what we have off the shelf. Those can be delivered within a couple of days to you. This is just some customer feedback on our mRNA IVT capabilities. We had a customer that used our from Georgia State University, used our mRNA for expression in cells with very good results. We had another customer in upscale medical university using our mCherry RNA for expression in primary cells. Another customer used our mRNA for in vivo work in mice with excellent results. Then we had some feedback from Merck and XAP using our mRNA synthesis, Merck for using our mRNA as antigen for membrane proteins that are difficult to produce, and XAP using just our custom mRNA synthesis, also with very good results. So we can deliver a lot of these services and products from discovery all the way to preclinical and clinical work. We have done many, for our CRISPR reagents, we have done over 100 GMP production batches. We have passed over 22 successful audits in different regions in the world. We have done 30 R&D projects, and we have supported our customer to achieve 6 R&D approvals. Here is just another reference from a customer that used our two-component sgRNA service and for human clinical trials with great success, and they were very satisfied from the results. Here is another example of our expertise in plasmid DNA services. We supported 2 companies, Temi RNA and Abogen, for development of their mRNA vaccines with our plasmid services. And we have a lot of expertise in developing plasmids for GMP plasmids. We have many years of experience. We have done 7 IND projects. We have done over 20 CMC projects. We have done many GMP batches and supported also mRNA customers with our plasmids. We have also complete capabilities for vector engineering and development. We can either produce the plasmids for AAV or lenti, or we can package the viruses themselves. We have full capabilities from research to GMP grade lentivirus service and AAV vector service also. And this is just a summary of our offerings, basically the key offerings that may be relevant to gene therapy development. You can see that we can deliver these services and products from research grade through preclinical grade all the way to GMP grade, antibody and protein expression, CRISPR, gRNA, and HDR templates, plasmid preparation. Our IVT mRNA GMP production is in development, should be available soon. Right now, we can deliver research grade and preclinical grade AAV vector service all the way from RUO to GMP grade, lentivirus service from RUO to GMP grade, peptide synthesis also. And here below, we have some products. Our cell isolation system is instrument for purifying cells like T cells, NK cells, stem cells for cell therapy development. We offer the instrument and the consumables related to it. We offer them as research grade, and we offer them as GMP grade. We also have excellent T cell activation reagent that we offer as a research grade and GMP grade. And as I mentioned, we have some other instrumentation for purifying plasmids and purifying proteins and antibodies that we offer for research use. Very convenient with magnetic bead purification and automation. In why choose GeneScript, we have a lot of expertise all the way from gene synthesis to plasmid production to virus production and CRISPR reagents production and services. We have worked with many customers around the world. We have delivered a lot of products and services. And basically, our customers have been successful in publishing very interesting work in a number of journals, some of them very high-profile journals like Cell, Nature, PNAs. We have over 80,000 journal references at this moment. And these are just some pictures from our GMP facilities. We have two GMP facilities in China, one in Nanjing and one in Zhenjiang, with very large capacity, with very diverse capabilities from all the way from plasmid production to viruses and antibody therapeutics and so on. And we recently opened a GMP site in the US, in Hopewell, New Jersey, 128,000 square feet. Right now, this site is capable of producing GMP plasmids, and very soon we'll be expanding the offerings to other modalities. And these are all the slides that I have for today, and thank you for your attention. I'm happy to answer any questions you may have.
Well, thank you very much, Rumen, for that informative presentation. We'll now begin the Q&A segment of the webinar, and we encourage you to continue submitting your questions for our speaker today. All right, let's go ahead and dive in here. Our first question, Rumen, is what is your AAV ITR guarantee?
Yeah, so we have a lot of expertise in developing and producing the AAV viruses. We have our own proprietary strains that we have engineered to provide higher ITR integrity. ITRs are very unstable. They can be degraded relatively quickly. So we have specialized AAV strains that can provide higher integrity. We have also developed our sequencing capabilities. ITR regions are pretty difficult to sequence because of very high GC content, but we have developed proprietary procedures for sequencing so we can achieve very clean and complete sequence of our ITRs. So we can basically produce them and we can show that they're intact in the end of the process.
Wonderful, thank you for that. Our next question is, what sgRNA libraries do you offer?
So we have a license and we offer a lot of the Broad Institute pre-validated gRNA library options, including Gecko libraries for genome-wide knockouts of human and mouse genes, or CRISPR synergistic activation mediators, some libraries for transcriptional activation of the genes in human and mouse. We also offer a lot of custom developed libraries. So as I mentioned, we have our Gene Titan technology that is very efficient at producing oligopools. And with these oligopools, we can develop many different mutant libraries, sgRNA libraries, and so on.
Great, thank you so much. We have time for just a couple more here. So let's move on to our next question. What is the timeline for your cell line development services?
Yeah, so we do a lot of cell line development work. Depending on the type of a cell line you require, if you are okay with cell pool cell lines, then the timeline is about 8 to 10 weeks. And if you require a monoclonal cell line, usually our timeline is from 14 to 18 weeks.
Great, thank you so much. Looks like we have time for just one last question here. Can you customize the LNP to target specific cell types or organs, for example, immune cells?
Yes, we have done quite a bit of development work internally. We have some pre-made formulations that can help support targeting specific cell type, for example, GERCAT cells, THP1 cells, and other cells or organs. Or we can work with you to develop custom formulations, depending on your needs.
Awesome. Well, thank you, Rumen, again for your time today and your important research. Any questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information used at registration. This webcast can be viewed on demand for 1 year until October 16th, 2025, and LabRoots will alert you via email when it is available for replay. We also encourage you to share that email with your colleagues who may have missed today's live event. Thank you to our audience for joining us today and to GenScript for sponsoring this webinar. Until next time, goodbye, everybody. Thank you, Rumen.
Thank you. Bye-bye.